Welcome to Brainstorm, where we give you a glimpse into the world of science for this Monday, April 30th, 2012. Our top story comes from the world of biotechnology. Researchers from the UK and elsewhere have made exciting progress in the development of synthetic genes. Regular DNA, as you may remember, is two strands of bases A, T, C, and G on a backbone of sugar and phosphate. The D standing for deoxyribose, which is a specific kind of sugar found in all life on Earth. However, that's not the only possibility. Scientists can synthesize molecules essentially identical to DNA but with different sugar in the backbone. Called XNAs, these synthetic molecules aren't new, for the first time can be transcribed from DNA and back. Transcription is normally a process that copies a segment of DNA into RNA and vice versa. So the researchers took the enzyme that performed this task and mutated them until they could work with XNA building blocks. Of course, this transcription process isn't perfect, but that's actually a good thing, because it allows mutations to occur in the XNA. That means when trying to obtain a specific function from XNA, they can use a process like evolution to get the best sequence. Separate researchers have also been able to make genetic material with a traditional DNA backbone but with different base pairs. If these two technologies were combined, it would result in a completely synthetic form of genetics. And as if that wasn't enough, XNAs may also give us insight into abiogenesis. The sugars that make up our DNA and RNA are actually quite complex, so it's entirely possible that the first genetic material used simpler molecules, like those found in certain kinds of XNA. Next, we turn to the world of medicine. Scientists at Duke University have used 3D modeling algorithms to produce a new potential treatment for cystic fibrosis. CF is a genetic condition which causes the malfunction of a protein involved with the water-salt balance inside the cell. This doesn't cause the protein to stop working entirely, but the defect makes the cell recycle the protein much faster. It's now understood that this water balance protein is misfolded in those with CF, but most drugs focus on slowing down the recycling process. In order to expand the number of potential treatments, the team at Duke had algorithms as the structure of the recycling protein looking for inhibitors. The top 11 compounds found by the simulation were then synthesized and tested directly with the protein. From that test, the best inhibitor was put in a culture of human cells with the cystic fibrosis mutation. It boosted the activity of the balanced protein by 12%, which is promising, but more testing is needed. And finally, a quick update from the world of biochemistry. Researchers in Israel may have developed a new way to protect people from chemical attacks, specifically a treatment against certain nerve agents that can be used by first responders and attack victims. Right now, the only protection from these chemicals are suits, gas masks, and other physical barriers that can be breached fairly easily. Similarly, people exposed to them are only given drugs to alleviate symptoms but not actually eliminate the chemical. So, the researchers took an enzyme found in humans called oxidase, which is involved with drug metabolism and detoxification. And although it does break down sarin and similar chemicals, it doesn't do so nearly well enough to counteract significant levels of exposure. Mutations were randomly made in the enzyme, and then bacteria were engineered to produce these variants. The bacteria were then exposed to the nerve agents to see which variant worked best. What resulted was an enzyme that worked up to 340 times better than the original found naturally in humans, making it a promising candidate for both preventative and post-exposure treatment for these kinds of chemical attacks. Well, hope you enjoyed this episode. If you did, please consider subscribing and be sure to check the links in the video description.